learners, greetings of the day. Welcome to SWAYAM courses on microbial technology, week number 6, lecture number 19. Myself, Devashish Sahu, currently working as research head, Bionaval Life Science Private Limited, Bengaluru, having more than 13 years of research experience in biological field. So, in today's session, we shall learn about an important aspect of metabolism and metabolic pathway that is quite essential to all the organisms and most relevant associated to energy generation cycle. But before we shall dive into the new topic, let us know and discuss the learning outcome of previous class, that is N-Dodorov pathway. So, the N-Dodorov pathway is a metabolic pathway that catabolizes glucose to pyruvate using a series of enzyme mediated reactions distinctive from those of more widely known as glycolysis and the PPP pathway or the pentose phosphate pathway. Named after Nathan Enter and Michael Dodorov, who elucidated its key steps in 1950s, this pathway is particularly prominent in certain bacteria and archaea. It provides an alternative route to glucose metabolism often employed by organisms that lack key glycolytic enzymes. The ED pathway is particularly significant in aerobic bacteria and archaea, including many gram-negative bacteria such as Pseudomonas and Zygomonas species. It is less efficient than glycolysis, yielding only one ATP molecule per glucose molecule compared to 2 in glycolysis. However, it provides flexibility for the organism to thrive in diverse environment, especially when the enzymes for glycolysis are lacking or are not active or less active. The production of NADPH in the ED pathway is another key aspect, as NADPH is crucial for anabolic reaction and the maintenance of redox balance within the cell. This is particularly advantageous for the organisms that require substantial amount of NADPH for biosynthetic processes. The ED pathway begins with glucose molecule, which is phosphorylated and thus oxidized to 6-phosphogluconate. The intermediate is then cleaved to produce pyruvate and glycerol dihydrate 3-phosphate, commonly called as G3P, which is converted into second pyruvate molecule via a glycolytic enzyme. The ED pathway differs in two significant ways from the EMP pathway. First, it generates NADPH, not NADH, from the oxidation of glucose 6-phosphate. This is used in many biosynthetic reactions. Secondly, only one triose phosphate is generated by the ED aldolase. As a result, only one ATP is generated per molecule of glucose rather than 2 generated by EMP pathway. The ED pathway provides a glycolytic route that bypasses the early step of EMP pathway that involves fructose derivatives. So, the takeaways of the ED pathway is EDP pathway or N-Dodorov pathway occurs in the cytoplasm of gram-negative bacteria, some gram-negative bacteria, some gram-positive bacteria and archaea. Such prokaryotes lack the critical enzymes required to carry out glycolysis. Some microorganisms carrying out the ED pathway are Pseudomonas, Azotobacter, Rhizobium, Agrobacterium, E. coli, Enterococcus, Zygomonas mobilis, Xanthomonas campestris, etc. These bacteria are aerobics due to low ATP yield per glucose. ED pathway is an alternative to glucose found in prokaryotes yielding 2 NADPH and 1 ATP per glucose molecule catabolized. So, in today's session, we will be talking about microbial photosynthesis, which is very relevant to each and every organisms, especially those who produce their own food, those are called autotrophs. So, let me introduce this topic with certain points. So, first we will have to discuss what is photosynthesis, a certain introduction to photosynthesis. Then we will introduce you to photosynthetic pigments, then compartmentalization and the importance of compartmentalization in photosynthetic pigments, and types of photosynthetic reactions occurring in microbes. So, you all know that photosynthesis occurs in plants. Microbial photosynthesis occurs in different microbes. So, it is as important to the plant. So, it is also important to the bacteria that carries about the photosynthesis. In general, photosynthesis is a biochemical process in which plant, algae and some bacteria convert light energy, usually from the sun, into chemical energy and stored in glucose form. So, it is just a type of sugar. So, this process is fundamental to life on earth, as it is the primary source of organic matter for nearly all the organisms, and it also releases oxygen into the atmosphere, which is essential for aerobic respiration. So, in photosynthesis, you will get 6 molecules of carbon dioxide, okay, and 6 molecules of water. So, these two will combine to form or be converted into 1 molecule of glucose and 6 molecules of oxygen using the light energy. So, photosynthesis is carried out by a diverse array of organisms, from microscopic algae, cyanobacteria to the towering tree of the forest. Autotrophs such as plants, algae and certain bacteria are capable of producing their own food through the process of photosynthesis. They use sunlight, carbon dioxide and water to synthesize glucose, which they can use as the source of energy. So, coming to the important photosynthetic microorganisms. So, photosynthetic microorganisms include a diverse group of organisms that uses photosynthesis to convert light energy into chemical energy. So, these microorganisms are critical component of various ecosystems and play a significant role in global carbon and oxygen cycles. The major type of photosynthetic microorganisms include cyanobacteria, algae, so include microalgae and macroalgae and certain protista. So, coming to cyanobacteria. So, cyanobacteria is also known as blue-green algae, commonly called as BGAs, are amongst the oldest photosynthetic organisms on earth. They are prokaryote, meaning they lack nucleus. They are found in wide range of environment, from ocean to freshwater to soil and extreme habitats like hot springs. So, pigments that is used for photosynthesis in cyanobacteria contain chlorophyll A, phycobiliproteins. So, phycobiliproteins, some types are phycocyanin and phycoerythrin, which gives them their blue-green color. The process they use is that they perform by oxygenic photosynthesis, producing oxygen as a byproduct that is similar to plants. What is the ecological and economic importance of this cyanobacteria? These are the primary producers. They contribute significantly to the primary production of aquatic ecosystem. Second, some cyanobacteria can fix atmospheric nitrogen, converting into a form usable by plants. Some of the biotechnological aspect is that they can be used in the production of biofuels, bioplastics and as nutritional supplement. Some example of nutrition supplement is spirulina. Then coming to the second organism that uses photosynthesis are called algae. Algae are eukaryotic organisms that can be unicellular or multicellular. Unicellulars are called microalgae, multicellular are called macroalgae. They lack specialized tissue found in higher plants. So, coming to the diversity, algae include green algae. So, green algae are alternatively called as chlorophyta, red algae called as rhodophyta, and brown algae called as phaeophyceae. Okay. These are the three differentiation: green algae, chlorophyta; red algae, rhodophyta; brown algae, phaeophyceae. Then what is the photosynthetic system in algae? So, basically the algae contains various pigments, chlorophylls, carotenoids and phycobiliproteins that allow them to capture the light energy efficiently. They perform as usual oxygenic photosynthesis that is similar to plants and cyanobacteria. Then what is the ecological and economic importance? The first importance is algae are crucial primary producers in marine and freshwater ecosystem, forming the base of food web. Second is biofuels. Microalgae can be explored for biofuel production due to their high lipid content. Third, nutrition and supplement. Algae are rich in vitamins, minerals and antioxidants, making them very valuable asset as a dietary supplement. Example is chlorella. Then bioremediation. Algae can be used to remove pollutants from wastewater. Then the third point, photosynthetic protista. So, they are often referred to as phytoplanktons. These are diverse group of eukaryotic microorganisms. They include dinoflagellates, diatoms and euglenoids. The common habitat of these protista are predominantly aquatic and are found in both marine as well as freshwater environment. Then the pigments. So, these organisms contain various pigments such as chlorophyll, fucoxanthin and peridinin. Like for other photosynthetic organisms, they perform oxygenic photosynthesis. Then ecological and economic importance of phytoplanktons are, they are considered as the primary producers. So, phytoplanktons are the primary producers in aquatic system, supporting many food webs. Carbon sequestration, they play a vital role in biological carbon pump, helping to sequester carbon dioxide from the atmosphere. Some photosynthetic protista, however, like dinoflagellates, can cause harmful algal blooms which can have significant ecological and economic impact. Then generally, if we are discussing about photosynthesis, we should discuss about photosynthetic pigments. So, photosynthetic pigments are very important for carrying out photosynthesis. Photosynthetic pigments are the molecules that absorb light energy used in photosynthesis. These pigments are crucial for capturing light energy and converting into chemical energy. Both plants and photosynthetic microorganisms such as algae and cyanobacteria rely on these pigments. So, there are three different types of photosynthetic pigment. So, these are broadly categorized into three main groups. Number 1 is chlorophyll, number 2 carotenoids, number 3 phycobiliproteins. First, coming to the chlorophyll. Chlorophylls contain chlorophyll A. So, chlorophyll A is a magnesium porphyrin complex with a long phytol tail which anchors to the thylakoid membrane. Function: it is primary pigment involved in light reaction of photosynthesis, absorbing light mainly in blue, violet and red wavelength. These are found in all photosynthetic organisms, that includes plant, algae and cyanobacteria. Chlorophyll B, similar to chlorophyll A, but with a slight difference in the side chain. A formyl group replaces a methyl group. So, the function expands to the range of the light absorption by capturing the light in blue and red orange wavelength and transferring the energy to chlorophyll A. Presence: it is found in plants and green algae. Now, discussing about chlorophyll C, chlorophyll D and chlorophyll F. These are the different variants of chlorophyll with different side chains. Basically, the function is adapted to absorb different light wavelengths. Chlorophyll C is found in brown algae and diatoms. Chlorophyll D is found in red algae and chlorophyll F found in cyanobacteria, that is living in low light environment. Then coming to another section, that is called carotenoids, or another family of pigment called as carotenoids. Carotenes, alternatively called as beta carotenes. So, the structure has hydrocarbon chains with alternating double bonds. They absorb light in blue and green region, protect against photooxidative damage by quenching reactive oxygen species. These are found in plants, algae and cyanobacteria. Xanthophylls. So, these are oxygenated derived of carotenes. They assist in light harvesting and photoprotection, involved in non-photochemical quenching, found in plants and algae. Phycobiliproteins. Phycobiliproteins are of two types: phycoerythrates, composed of protein complex with covalently attached bilin chromophores. They function as they absorb the light in yellow orange region and transfer the energy to chlorophyll A. They are found in red algae and cyanobacteria. Now, another component is phycocyanin. These are similar to phycoerythrin, but absorb light in the orange red region. They capture light energy and transport it to chlorophyll A, and they are found in cyanobacteria and some red algae. Then another category is allophycocyanin. This is another phycobiliprotein that bridges energy transfer from phycocyanin to chlorophyll A. They absorb light in the red region, commonly found in cyanobacteria and some red algae. The function and mechanism. Let us discuss about the function and mechanism of photosynthetic pigments. So, photosynthetic pigments play several critical roles in photosynthesis. The first and the foremost important activity of these photosynthetic pigments is the light absorption. So, these pigments absorb light energy which excites the electron to a higher energy state. Chlorophylls primarily absorb light in the blue and red wavelength, while carotenoids and phycobiliproteins extend the range of light absorption by capturing in additional wavelengths. Energy transfer. The absorbed light energy is transferred between pigments within the photosystems. There are two photosystems, PS1 and PS2, until it reaches the reaction center, where it is used to drive the photochemical reaction that produces ATP and NADPH. Third one is photoprotection. Carotenoids protect the photosynthetic apparatus from damage by reactive oxygen species and excessive light by dissipating excess energy as heat, that is non-photochemical quenching. Photosynthetic pigment in plants, higher plants. So, chloroplasts are there. So, contains chlorophyll A, chlorophyll B and carotenoids. In thylakoid membrane, this is the site of light absorption and energy conversion process. So, what is the function? So, both chlorophyll A and chlorophyll B capture light energy and drive the light dependent reactions. What is the use of carotenoids? They extend the range of light absorption and provide photoprotection. Then photosynthetic pigments in microbes. First is cyanobacteria. The pigments that are present in cyanobacteria are chlorophyll A, phycobiliproteins. Phycobiliproteins contains phycoerythrin, phycocyanin and allophycocyanin and carotenoids. So, the adaptation for cyanobacteria is that they can thrive in various light conditions due to the diversity of pigments. Then coming to the algae, green algae contains chlorophyll A and B similar to higher plants. Red algae contains chlorophyll A, D and phycobiliproteins adapted for low light conditions in deep water. Brown algae and diatoms contain chlorophyll A, chlorophyll C and fucoxanthins, a carotenoids that is adapted for different light environment in marine ecosystem. Then protista, example are dinoflagellates and diatoms. So, the pigment may vary widely. This may include chlorophyll A and chlorophyll C and different carotenoids like pyridinin and fucoxanthin. The function is to adapt to capture light efficiently in diverse aquatic environment. The compartmentalization, let us discuss about compartmentalization, which is another very important aspect. The compartmentalization of photosynthetic pigment in plants and photosynthetic microbes is a fundamental aspect of their ability to perform photosynthesis efficiently. This organization allows for optimal light absorption, energy transfer and photoprotection, ultimately driving the conversion of light energy into chemical energy. In higher plants, the organization of thylakoid membranes into grana and stomatal thylakoids allow for optimal light absorption and energy transfer. Algae exhibit a wide area of chloroplast structure reflecting their adaptation to different light environment. Cyanobacteria, despite lacking membrane bound organelles, have specialized structure like phycobilisomes that facilitate efficient light capture and energy transfer. Understanding this compartmentalization of photosynthetic pigment provide an insight into the adaptability and efficiency of photosynthesis in various organisms and environment. So, photosynthetic pigments in higher plants, first we start with the chloroplast structure. They contain thylakoid membranes. These thylakoid membranes are the primary sites of photosynthesis within the chloroplast. These membranes form a series of flattened sacs or called as thylakoids, which are often stacked into structures called grana. The thylakoid membranes are interconnected by stomatal thylakoids or called as lamellae. Then compartmentalization of pigments in photosystem 2 and photosystem 1. So, in photosystem 2, locally primarily in the grana, photosystem 2 contains chlorophyll A, chlorophyll B and various carotenoids. The light harvesting complex 2 associated with photosystem 2 captures light and transfer it to the reaction center. Photosystem 1 or alternatively called as PS1 located mainly in the stomatal thylakoid and the edge of grana stacks. PS1 also contain chlorophyll A, chlorophyll B and carotenoids. The light harvesting complex LHC1 of photosystem 1 capture light energy and transfer it to the reaction center. Then ATP synthase and cytochrome B6 complex. These components are distributed throughout the thylakoid membrane, participating in the electron transport chain and ATP synthesis. What is the functional significance? Separation of PS1 and PS2. The spatial separation of PS1 and PS2 optimizes the flow of electrons and reduces energy loss during the electron transfer. It also organizes the efficient light capture. So, basically this organization of the pigment within this LHCs maximize the light absorption and energy transfer efficiently. Third point, most important point is that photoprotection. Carotenoids in the thylakoid membrane protect against photooxidative damage by dissipating excess energy as heat. So, photosynthetic pigment in algae, chloroplast diversity. Algae exhibit wide variety of chloroplast structures reflecting their adaptation to different light environment. Green algae alternatively called as chlorophyta. So, the structure present in the higher plants, the chloroplast structure present in the higher plants. So, here the thylakoid membranes are organized in grana and stromatal thylakoids. The pigment is as similar to the plants. In the chlorophyta, they contain chlorophyll A, chlorophyll B and carotenoids. Red algae called as rhodophyta. So, the chlorophyll structure or the chloroplast structure, here the thylakoids are not organized into grana stacks, but are instead universally or uniformly distributed. The pigments that they contain are chlorophyll A, phycobiliproteins, alternatively some are phycoerythrin and phycocyanin and carotenoids. Compartmentalization is that phycobilisomes, the complex of phycobiliproteins, are attached to the surface of the thylakoid membrane, optimizing light capture in deep water and low light environment. Brown algae and diatoms called as Phaeophyceae and Basilorophyceae. The thylakoids are often grouped into band of 3. They contain chlorophyll A, chlorophyll C and carotenoids phycoxanthin that give them their characteristics brown color. Compartmentalization. The unique arrangement of thylakoid and pigment allow these algae to capture light efficiently in various aquatic environment. Type of photosynthetic cycles in microbes. The key photosynthetic cycle in microbes include oxygenic photosynthesis occurring in cyanobacteria and algae, anoxygenic photosynthesis occur in purple and green sulfur bacteria and photoheterotrophs. Oxygenic photosynthesis are similar to the plants, is carried out by cyanobacteria and eukaryotic algae. It involves the splitting of water molecules to produce oxygen and follows the same fundamental process as in plant photosynthesis. The light dependent reactions photosystem 2, light energy excites the electron in chlorophyll, splitting water molecule to release oxygen, protons and electrons. The oxygen is released at bioproduct. Photosystem 1, here the photons which are absorbed by chlorophyll re-excites the electron. These re-excited electrons are then used to reduce NADP+ to NADPH. However, here what happens? The proton gradient drives the ATP synthesis via ATP synthase, producing ATP from ADP and inorganic phosphate. Another part of the oxygenic photosynthesis is light dependent reaction. Carbon fixation, CO2 is fixed to ribulose 1,5-biphosphate by enzyme ribulose 1,5-biphosphate carboxylase into 3-phosphoglycerate. Reduction phase is the ATP and NADPH from the light dependent reaction in presence of photoglycerate kinase and glyceraldehyde 3-phosphate dehydrogenase enzyme reduce 3-phosphoglycerate to glyceraldehyde 3-phosphate. So, here what happens? There is regeneration of glucose G3P is used to regenerate ribulose 1,5-bisphosphate, enabling the cycle to continue. Then coming to anoxygenic photosynthesis. Anoxygenic photosynthesis occur in several groups of bacteria including purple bacteria, green sulfur bacteria and heliobacteria. These microbes do not produce oxygen as they use the electron donors, other electron donors instead of water. So, some examples are purple bacteria and green sulfur bacteria and heliobacteria. So, the third type is photoheterotropy. Photoheterotropic microorganisms uses light to generate ATP, but obtain carbon from organic compounds rather than fixing CO2. This process is a combination of photoheterotropy and heterotropy. Some examples are purple non-sulfur bacteria, green non-sulfur bacteria. Then coming to the summary, we have learned about photosynthetic cycles. So, photosynthetic cycle in microorganisms show remarkable diversity in their mechanism and adaptability to different environmental conditions. Oxygenic photosynthesis in cyanobacteria and algae resemble that in plants produce oxygen using both photosystem 1 and photosystem 2. Unoxygenic photosynthesis in purple bacteria, green sulfur bacteria, heliobacteria involve different electron donors and photosynthesis adapted to anaerobic environment. Photoheterotrophs microbes blend phototropics and heterotropic process using light to generate ATP while relying on organic compound for carbon. Understanding these cycles highlights the versatility of photosynthesis beyond the plant kingdom, contributing to the productivity and ecological role of microbial community. So, the topics that is covered in the next lecture is the nitrogen fixation. So, microbial nitrogen fixation is a critical biological process which certain microorganisms convert atmospheric nitrogen into ammonia, making nitrogen accessible to living organisms. So, this process is a cornerstone of nitrogen cycle which is essential for biosynthesis of proteins, nucleic acid and other cellular components. So, here the learner also have the opportunity to understand the mechanism, genetics, regulatory aspect of nitrogen fixing genes present in the microbe along with the importance of nitrogen fixation by microbes in ecology, agriculture, environment and industry. I am excited to share this information with you and looking forward to our discussion. Thank you for joining this session.